Hi everyone, I'm Tabby, and today I'm going to be talking about investigating the evolutionary relationships of the pantropical moth genus Parasa. This is part of my PhD project, so I'm in my second year of my PhD and I'm based at the University of Oxford and I'm funded by the African Natural History Research Trust. So in general, we expect to see a congruence between biogeography and taxonomy, whereby higher taxa, so orders and families, have a broader distribution, whilst genera and species would have a more confined distribution with local adaptations. So this is the kind of relationship we would expect to see given the geological history of the Earth. However, this is not always the case, and sometimes we observe global disjunct distributions for a single genus. This has especially been noticed in pantropical Lepidoptera. So there are some examples of true pantropical butterfly genera, such as Leptotes, Vanessa, and Hyperlimnus. And there's been recent phylogenetic evidence supporting their evolutionary relationships. However, in some cases, inaccurate taxonomy exists. For example, the widespread butterfly genus Eurema was recovered as polyphyletic in a multi-gene phylogeny. And so the taxonomy was recently amended to include 2 additional genus names, and such incorrect classifications of widespread genera may still exist and require investigation. So my project is studying a single genus of moth within the family Limacodidae. It's called Parasa, and they're found throughout the global tropics. There are around 100 to 200 species in the genus and related genera, which I will discuss shortly. So here you can see 3 examples of Parasa. So there's Chloris from the Americas, Ananii from the Afrotropics, and Lepida from Asia. And throughout, I'll be referring to these 3 main tropical regions. As you can see, they share the same phenotype of brown and green banding on the forewings. And it's really important to note here that the type species of the genus is Parasa chloris, which is found from North and Central America, as this has taxonomic implications should the genus require major revision. So here we can see a very rough distribution of Parasa throughout the global tropics. And just from our knowledge of these areas, it appears that Parasa are mainly present in forests. This may or may not relate to adaptation of their green and brown colouring. Not much is really known about their life history or behaviour, but Parasa species are considered to be generalists and their caterpillars feed on a wide variety of food plants. Some species are considered pests of agricultural crops in Asia and Western Africa as well. So finally, to add some taxonomic confusion, Parasa has a rather convoluted history, and there are currently many other genera also associated with Parasa due to morphological similarities. For example, these 3 lima-coded genera here, so Euclea from the Americas, Latoya from the Afrotropics, and Thespia from Asia, are considered to be closely related to Parasa as they also have some green and brown on their wings. And this conclusion is supported by morphology only, and it's never received any genetic support. So this group containing Parasa and the associated genera is thus referred to as the Parasa complex. Throughout the literature, taxonomists have questioned the monophyly of Parasa and identified that this is potentially an issue. However, no attempt has ever been made to investigate its global evolutionary relationships or to stabilize the nomenclature. So the overall aim of my PhD is to build a large global phylogeny of Parasa and its associated genera to try and infer these evolutionary relationships. So are global Parasa species all closely related, or have they just convergently evolved to share this phenotype? And this can really help us to set taxonomic limits for the genus and discover whether major changes need to be made to the naming. So for this, I am going to be utilising Natural History Museum collections, which hold a wealth of biological information to gather specimens for genetic and morphological investigations. So there are 3 main museum collections that I visited with specimen holdings from the 3 tropical regions. I will then extract DNA and sequence whole genomes, which is now relatively very cheap, and then select nuclear genes to build a phylogeny. As I've not yet completed all of my sequencing, the genetic results I'm presenting today will be just based on the CO1 barcode. So sequences for members of the Parasa complex were already available on the Barcode of Life database or very kindly shared with me for this project. So CO1 barcodes were available for 46 taxa in the Parasa complex, plus an outgroup as well. To aid the building of the phylogenies based on only the short CO1 sequence, additional evidence can be found from morphological characters. So synapomorphies inform us of a common evolutionary history, and often combining DNA with morphology can yield more resolved trees and better supported groupings. So to build a morphological character matrix, I focused only on structural characters of the male genitalia and the antenna. I chose not to include characters regarding coloration or wing pattern. As Parasa generally share this phenotype, this would immediately introduce homoplaty into the dataset. So in total, I coded 41 characters for 58 in-group taxa and 1 outgroup. To build the trees, I ran 3 types of analyses. So one included all of the taxa, one removed some American genera, and one also removed some Asian genera. And the taxa that I selected to be removed either had very obvious morphological differences to Parasa and or had unstable positions throughout the previous analyses. So I just wanted to see whether removing them would improve the resolution of the tree. And then for each of these analyses, I also used multiple phylogenetic methods. So the DNA-only trees were built by running both Bayesian inference and maximum likelihood. Morphology-only trees were built using maximum parsimony in TNT, and they were both unweighted and then weighted against homoplasy. And finally, combined DNA and morphology data were analysed using Bayesian inference. So in total, I had over 20 different phylogenetic trees. So here I'm just going to present the most well-resolved tree and talk through what it shows. So this is the analysis that produced the best tree I'm going to be talking about. So it's the third analysis type with selected American and Asian genera removed, looking at DNA only and analysed through Bayesian inference. So this is the resulting phylogenetic tree from that analysis. So I'm firstly just going to talk about what conclusions we can possibly draw. So the first and perhaps most important thing to note that in this analysis, as well as in all of the other ones that I ran, Parasa was not recovered as monophyletic. So hopefully you can see some of the genus names along here. As a reminder, these are other genera that are considered members of the Parasa complex, but there's never really been an attempt to distinguish what this really means in terms of relatedness. In addition, all of these branch lengths are pretty long. So this scale bar shows a 9% difference in CO1 sequence. So perhaps all of the things on here are actually really quite different. We could be looking at potentially lots of different genera here. But with this, I want to point out where Parasa chloris is, which is the type species of the genus. So chloris is found here, meaning that this clade at the very least is true Parasa. In every analysis, including the morphological only, chloris was found to be sister species to Viridogrissia, which is another American species, so they clearly share some affinities. This is what both of those species look like. And despite looking pretty similar, I think this really emphasizes the importance of not just considering wing color when we're placing species in the same genus. From the phylogenetic results, these two are clearly very closely related, while species that look considerably more like chloris with the green and brown forewing banding are less similar in the DNA. So we just need to make sure that we are using integrative taxonomy when we're placing species in a certain genus. When considering biogeography, the 3 tropical regions are here corresponding with the colours of the clusters. I hope that's clear for everyone. We can firstly see that the regions don't necessarily group nicely together, and that there are some cases where taxa from different regions are clustered together. And these relationships were also found in all of the other analysis types. Whilst adding more taxa could make these relationships a little bit clearer, perhaps this is an indication of multiple colonizations of the different regions. If we look at the oldest split here and even the next couple after this, we can infer an African origin as denoted by the green clusters up here. The out of Africa hypothesis is well founded and has support in a lot of other groups, including butterflies like Hyperlimnus and Caraxes. But to infer colonization routes, I would ultimately like to date the tree somehow to include divergent estimates as well. So finally, I'll just touch on what is still unknown. As you can see here, there is a large polytomy on the tree, which suggests that the placement of these taxa and clades is not super clear yet. I was also a bit surprised to find that the analysis that combined morphology and DNA did not form a super well-resolved tree. It had an even larger polytomy with all of these smaller clusters coming off. Although it did improve the support values of clusters that have, there's a few here with little stars next to them. That's where morphology did actually help the support values there. So in general, Limacodids have very simple genitalia, and so perhaps the characters that I chose to code for were not super informative when we're considering a really large global scale. So to properly solve this relationship, ultimately I need more gene sequences and probably some more taxa as well. I aim to extract and sequence more DNA in the new year, which I'm very excited about. And with these results, I can then start to look into deeper biogeographical patterns with things like dispersal vicariance analysis, divergence times, and if the sequences are good enough, I can consider gene evolution of certain traits as well. So with that, I'd like to thank my two supervisors, Tim and Sonia, as well as Marianne Espeland and my funders at ANHRT. And thank you to all the organisers, and thank you all for listening.